this is Adil. Today I am going to talk about another histopathological finding, that is acantholysis. Acantholysis is a separation of keratinocytes due to defect in desmosomes. Here you can see two keratinocytes, and between two keratinocytes there are desmosomes. If there is any damage or any defect in these desmosomes or desmosomal proteins, that lead to separation of two keratinocytes. Here you can see the separated keratinocytes, and the separated keratinocytes are known as acanthocytes. Acanthocytes, which are also known as acantholytic cells. Acantholytic cells. Let's see clinical features of acantholysis. The acanthocytes are oval-shaped cells, and the nucleus occupies 7/8 portion of the cell. Here you can see. And the perinuclear halo is seen due to the condensed cytoplasm. Here you can see a condensed cytoplasm. To see these acanthocytes, generally a cytological test is done which is known as Zhang smear. If a patient comes with blisters, first you will rupture the blister and then take scraping from the base of the blister, then prepare smear, then stain it with gisma and right staining. Finally, examine this smear under the microscope. If under microscope you see oval-shaped cells, nucleus occupies 7/8 portion of cell, and perinuclear halo, then you will label those cells as acanthocytes. There are other cells which can be seen by Zhang smear, like multinucleated giant cells, dyskeratotic keratinocytes, necrotic keratinocytes. These acanthocytes are seen in pemphigus, and multinucleated giant cells are seen in viral infections like herpes and chickenpox. Dyskeratotic keratinocytes are seen in Darier's disease and also in Hailey-Hailey disease. And necrotic keratinocytes are seen in epidermal necrosis. And this epidermal necrosis can be seen in Steven-Johnson syndrome and toxic epidermal necrolysis. Now let's move on to various conditions and their defects where acantholysis is seen. The first example which I am taking here is pemphigus vulgaris. In pemphigus vulgaris, desmoglein 3 is more defective than desmoglein 1. In pemphigus foliaceus, desmoglein 1 is defective, and in paraneoplastic disease, plaquin family is defective, which include desmoplaquin, placoglobin, and envoplaquin. Darier's disease and Hailey-Hailey disease are due to defect of calcium ATPase. You might be wondering why calcium ATPase defect lead to acantholysis. In my earlier videos, I have explained that desmosomes are also known as calcium dependent adherent molecules. It means desmosomes are dependent on calcium. If their calcium ATPases are defective, then desmosomes also become defective. When desmosomes become defective, finally the acantholysis will occur. Subcorneal pustular dermatosis is also known as Sneddon-Wilkinson disease. It is due to defect in desmocolin. In all these conditions, intraepidermal disorder is seen. What is intraepidermal disorder? In intraepidermal disorder, keratinocytes get separated, that is acantholysis. Whenever the keratinocytes get separated, a gap is formed, and inside the gap, extracellular fluid accumulates. When extracellular fluid accumulates in this gap, there is the formation of intraepidermal blister. Hope you have enjoyed this video. Thank you.